Okay, so the last video, revision video for the immune system is just taking a quick look at how antibodies destroy pathogens. So remember, which cell are antibodies released from? That is the plasma cells. So plasma cells release an antibody with a antigen binding site that is specific for a specific antigen, so it fits exactly only one antigen. So how do they destroy pathogens? So I'm giving you two clues here in terms of images. If you want to try and answer that, pause and return and I'll explain. Okay, so the first one, you'll notice that there are several antibodies and they obviously have two antigen binding sites per antibody, so they can bind two pathogens, and you notice that each of them is bound to two pathogens and they're sort of clumping together. If the pathogen is clumped together like that and held in this structure, those pathogens obviously can't infect cells and then cause disease. So that is a way that antibodies will help prevent a pathogen from causing disease by gathering them together in these clumps. That process is called agglutination. So that is the key word you need to be using when you're describing that process of how antibodies help destroy pathogens or prevent pathogens from causing disease. So that's one. The second thing is that obviously the pathogen will have probably several antigens on its surface, many thousands most possibly. All of those are potential binding sites for an antibody. So you can have a situation where you may actually have several antibodies bound. Just move that. You may actually just even have one. That's fine. What that does is it marks the pathogen for phagocytosis by phagocytes. So it enhances phagocytosis of that pathogen. So let us very quickly draw a quick phagocyte. So this is obviously not to scale. The antibodies will be a lot smaller and the phagocyte will then be, this is a rather scary big phagocyte doing its job. And there's a nucleus. And it is engulfing, remember that's the word, engulfing or ingesting the pathogen and those antibodies by phagocytosis, and it will then, that will create a phagosome. Lysosomes will fuse with a phagosome, and the lysosomes will release their enzymes, lysozymes, into the phagosome and hydrolyze the pathogen. So there is a specific name for this. You don't need to know. Let me just describe it. So enhances phagocytosis of pathogen by binding. So that's what the antibody does. There is a specific name if you really like to know. It is called, do you know? I'll be impressed. Opsonization. 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 Can be, I think it might be spelled with a Z. I went for the UK standard spelling with an S, but Z would be perfectly acceptable as well. So agglutination, gathering a pathogen together, and opsonization, enhancing phagocytosis of a pathogen, are the ways that antibodies destroy pathogens. And that is it. Thank you.